sporogenesis. Sporogenesis. During the development of microsporangium, during the development of microsporangium, the cells of sporogenous tissues, the cells of sporogenous tissues may divide in various planes, and finally separate from each other to function as microsporocytes, or microspore mother cell, Some of microsporocytes degenerate and provide nourishment to others. The surviving microsporocytes are concerned with each other by cytoplasmic interconnection, and have prominent diploid nuclei Each microsporocyte then develop an inner layer of chylage, which breaks the cytoplasmic interconnection, with other microsporocyte. The separated microsporocytes then divide. The separated microsporocyte then divide by meiosis and give rise tetrads of and give rise tetrads of haploid microspores. Haploid microspores by the process called cytokinesis. By the process called cytokinesis. Cytokinesis results in the separation of cytokinesis results in the separation of 4 haploid nuclei. 4 haploid nuclei into 4 separate cells. Into 4 separate cells which are arranged in tetrad. Which are arranged in tetrads and give rise threads of haploid microspore and give rise threads of haploid microspores. Usually the arrangement of microspore in a tetrad is tetrahedral During the microsporogenesis, the cells of tapetum during the microsporogenesis, the cells of tapetum provide various enzymes. Provides various enzyme, hormones, amino acid, amino acid and other nutritive material and other nutritive material to the dividing microsporocytes, to the dividing microsporocytes. In angiosperm, the cells of tapetum are of two types: glandular tapetum, the cells of which secrete substances and finally break down at the time of pollen maturation. Second, periplasmodial tapetum, the cells of which break down early by dissolution of their walls so that tapetal protoplast form a multinucleate protoplasm. Now we will see a structure of pollen grain. Structure of pollen, microspores are and pollen grains. Microspores are pollen grains are haploid, uninucleate, are haploid, uninucleate. Minute spore produced in minute spores produced in large number as a result of meiosis in microspore mother cell, microspore mother cell inside the microsporangia. Inside the microsporangia. Each pollen grain of angiosperm, each pollen grain of angiosperm has two cells, Number first, vegetative cell, and second, germinative cell. And germinative cell. The pollen grains are generally globose in shape. The pollen grains are generally globose in shape. Though several others are also found in oval. The term sporoderm is usually used to designate collectively the two major wall layers. The term sporoderm. The term sporoderm is usually used to designate collectively pollinate collectively. The two major wall layer. The two major wall layer. First is exine, which is outer layer, and chiefly composed of sporopollen and chiefly composed of sporopollen. Second layer is intine, which is inner layer, and chiefly composed of pectocellulose and chiefly composed of pectocellulose. In angiosperm, pollen grains the exine is mainly derived from tapetal substances during sporogenesis, and intine is derived from male gametophyte, during development. सो फ्रेंड्स, इस वीडियो लिंक में इतना ही। अगले वीडियो लिंक में हम लोग डेवलपमेंट ऑफ मेल गैमेटोफाइट के बारे में अध्ययन करेंगे। तो आप हमारे अगले वीडियोज़ की प्रतीक्षा करें। धन्यवाद।